Hello, everyone, and welcome to today's webinar, Microarrays, an essential tool for predictive genomics. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues you have if you have any trouble seeing or hearing the presentation. Now, in addition to answering your questions, we have some questions for you. Please see the survey window at the left of your screen. And just below that, you will see a list of resources to further your predictive genomics journey. Be sure to check them out. And with that, I'd like to now welcome our speaker, Dr. Steve Jackson, Senior Member Applications Development at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Presenter tab at the bottom of your screen. Steve, welcome. You may now begin your presentation.
Hello, my name is Steve Jackson, and I'm the Associate Director of Product Applications at Thermo Fisher Scientific. In today's webinar, I'm going to introduce to you predictive genomics and how the applied biosystems microarrays are essential for having good outcomes from predictive genomics research. During the talk, I'll introduce the concept of predictive genomics. I'll introduce to you some of the biobanking initiatives that make use of predictive genomics for researching health risks in populations. I'll talk about pharmacogenomics a little bit, and I'll tell you why modern microarrays are the tool of choice for predictive genomics research. And then finally, I'll summarize with a bit of conclusions. So we all know that global healthcare costs continue to rise. Overall, global healthcare costs as a percent of GDP around 10%. The estimated loss to the global economy due to chronic illnesses is estimated to be about $47 trillion by 2030. And one of the leading causes of death worldwide is the adverse effect of drug reactions, or ADRs. Now, I'll mention that some of this data is kind of old, and in fact, that the actual costs and some of these numbers might be a little bit different now. But what is absolutely clear is that healthcare costs need to be controlled and need to be understood better, especially in light of all the genomic information that is becoming available to us since the Human Genome Project has finished. So making use of this data will hopefully help reduce some of these costs and bring better outcomes for people. So predictive genomics is a way to take this genomic information and try to make use of what is known about the human genome, what is known about the demographics, and make predictions about disease risk, a response to a drug, carrier screenings, and other things that might impact health costs and health outcomes. So it's a capability that will predict diseases, help understand disease risk, understand drug responses, and again, will help us manage healthcare costs and outcomes for patients. One of the easiest ways to understand predictive risk is to understand about rare inherited disease disorders. These are typically Mendelian diseases. Things are inherited in Mendelian or semi-Mendelian fashion. These are fairly easily understood and explained because there's a single gene, monogenic or at most maybe a handful of genes, which contribute to the phenotype or the pathology. Now, in spite of the fact that they're well understood or easy to explain, a very small fraction of the overall population is affected by these types of disorders. And therefore, they don't have a tremendous risk on the overall health costs of communities. On the other hand, common diseases such as diabetes, cardiovascular diseases, cancers, these are often the interplay of a large number of different genes and are known as polygenic diseases. It's very difficult to fully analyze or predict the phenotypes of these just by studying these individual genes on their own, because the way they interact with each other, the way they interplay with each other and the environment and with the demographics can be extraordinarily complex. Having said that, these diseases make up over 5% of the types of diseases that are affecting people. And therefore, there's a very large effort to understand the causes of these diseases, to be able to predict risks for these diseases, and to help understand treatments for these diseases. So a number of different countries and health organizations have undertaken vast projects to understand the genomes present in their populations or in their communities. One of the best examples of these is the UK Biobank. This is a project that was started about 20 some odd years ago by the United Kingdom. And it was an effort to collect demographic information, biochemical information, phenotypic information, and genetic information from at least 500,000 individuals across the United Kingdom. This is a multi-year project that has been ongoing, and they have accumulated a vast amount of data on the genetics, the metabolomics, the demographics, the phenotypes, the growth characteristics, as much related information that they can on these individuals, and put them into a publicly accessible database. This UK Biobank is a phenomenal source of information for researchers. And you can see from the graph on the left that the number of publications that reference the UK Biobank was slow starting. And of course, this is when they were in the process of defining the project and collecting samples. But since about 2017 or so, it has undergone an exponential increase in the number of scientists, the number of publications that are making use or referring to data that is in the UK Biobank. We'll talk about some examples about some of the research that has successfully come out of this project. But it has really helped us understand the link between people's environments, their demographics, their metabolic rates, and their genotypes. The graph on the right shows the example of what's known about the number of metabolites versus the number of SNPs. And one of the conclusions that came out of the study was that the interaction between the genome and the phenotype is very complex. For example, for some of the for example, if you look at if there are 100 metabolites are affected by SNPs that are in 30 different loci. Some of the other metabolites are affected by loci that are in the hundreds of different loci. So the interplay between the genome and the metabolome is very complex, making use of the data that has come out of the UK Biobank. So as I mentioned before, one of the goals of these types of projects is to help understand healthcare costs and help people that are within these communities. So as the UK Biobank was starting to accumulate data, they were finding that approximately 22% of the individuals in this biobank had at least a threefold increase in risk for a common disease. They had genotypes that were known to be associated with cardiovascular diabetes or other complex diseases. So that's a large number of people that were at risk. And if you look at the health costs of some of these diseases, that the UK spends about 29 billion pounds on cardiovascular diseases, 6 billion on cancer, 10 billion on diabetes. And so being able to reduce risk in these diseases would vastly help the UK control their healthcare costs, not to mention help the individuals that might be afflicted with these syndromes. So there are a couple of examples that I've pulled out from the UK Biobank data showing specifically how investigators are making use of this data. On the left-hand side of this slide is results from a paper that came out in 2019, where they looked at 16,000 individuals in the UK Biobank cohort that were afflicted with myocardial infarction or other kinds of heart failures. They were specifically looking for loci that were associated with left ventricle dysfunction. And they came up with a large number of different loci indicated here with the genes that are in red or blue that are specifically affecting different aspects of left ventricle heart function. So using this now, they can now come up with some sense of the risk of individuals that contain alleles of these risk loci and help manage the way that they either have lifestyle or diet or something like that to help reduce the possibility of there being myocardial infarction or other heart dysfunction. On the right-hand side is a different study where investigators were mapping the link between the metabolome, the genome, and specific syndromes specifically. And it's a very complicated figure, which I'm not going to spend a lot of time going into, but the reference does a very good job about explaining the way that The link between some of these loci that are identified in the cohort that produce changes in metabolites and risk to particular diseases was possible to analyze. Excuse me. And again, this helps the UK scientists understand how people that have particular alleles might be at risk for either metabolic dysfunction or disease function or both. So based on this model, when it was starting out, there were a lot of other organizations that wanted to build off their findings, extend their findings, and make use of their methods for understanding their own populations. One of the groups that did this is the Million Veterans Program. This is based off of the United States Veterans Administration program. The VA health system is responsible for maintaining the health of all the veterans that have served in the U.S. Armed Forces. And therefore, it's a very large database of, again, phenotypic, metabolomic, demographic information from U.S. veterans. They collected samples from a large number of them. They're going on 1 million different samples. On the left-hand side, you can see that there is some indication about the demographic distribution of people in this in the Million Veteran Programs sample database. One of the benefits of this program is that it spans across a large number of different ethnic varieties that are found in the United States: African American, East Asian heritage, Hispanic heritage, white heritage. It spans a very wide range of ethnicities, and therefore gives a pretty complete sampling of people in the U.S. Building off of this data, the Million Veterans Program scientists have begun to dissect the data, and in this one paper that's highlighted on the right-hand side of the slide, you can see that they have identified a couple of loci that are identified with suicidal thoughts among veterans. And so again, this will help them identify people that might be at risk for suicidal thoughts, and might help lead to interventions before those can be acted upon. Now, one of the other things that we're learning as we're analyzing a large number of different ethnicities and different genetic backgrounds is that the different sample collections might not be applicable across all of the different ethnicities in the world. For example, it's known that the UK Biobank is highly selected for Northern European populations, but it might not be appropriate for all other populations. And the example on the left is from a paper that was published looking at the FinnGen data. Finland is introducing a similar sort of program looking at the demographic and genetic information for Finnish populations. And this is interesting because Finland and by extension Estonia has been a somewhat isolated population with higher sorts of interbreeding, and more isolated mixing. And so they might have a different distribution of alleles that lead to specific phenotypes than UK Biobank or other, like, for example, African populations. And so on the left, what you see is that the distribution of samples and frequencies of people with diabetes, atrial defibrillation, and other syndromes are different in Finnish populations, shown in blue, compared to the UK Biobank populations, shown in red. Type 2 diabetes is much more prevalent in Finland than it is in UK Biobank samples. Estonia has undergone a similar population or a similar program for similar reasons. Estonia was isolated, and therefore the allele frequencies and diseases present in Estonia might be slightly different. And indeed, what you can see is that asthma is especially prevalent in Estonian populations compared to Finland or the UK population. Atopic dermatitis is another one. So what is clear is that having a data set that is specific for your population will be of better use for understanding and making use of the genetic diversity in your population and making risk scores more appropriate for your own communities. Another example shown on the right-hand side of this slide is from the UK Biobank, showing that if you break down the different ethnicities in the UK Biobank, you can see that the allele frequencies vary by quite a bit. For example, there's this one allele of apolipoprotein B, which is more common in African populations than in other populations. Some of these other ones are more common in Southeast Asian populations. So again, there are different allele frequencies present in different populations, and presumably that would lead to different risks. And so having a specific way to analyze the genotypes of your own populations would lead to more specific healthcare and risk decisions. Because of this, the Taiwan government has undertaken a very similar program. This is the Taiwan Precision Medicine Initiative. And in this case, they're doing a similar sort of thing. They're researching the ways to incorporate predictive genomics into routine clinical care for ethnically Chinese Han and also Taiwan and indigenous populations as well. Currently, they have 16 medical centers that are enrolled in providing samples for this study. It's hoping by 2024 they will have over 1 million participants. And the idea is that they would continue to collect genomes. They will make use of a custom array, and I'll also have more to say about that in a second. Profile the participants, analyze the data, and hopefully they will use this data to identify risk alleles and understand, the way that the genotypes and ethnic backgrounds are contributing to the different health decisions that we made in the Taiwanese populations. So another aspect of predictive genomics is pharmacogenomics. This is a field of study which builds off of the understanding of the way that individuals process drugs in their body. This can make use of either removing drugs once they're used, the way that the drugs are cleared from your system, the way that drugs might be modified to convert them from a precursor to an active drug, or the way that drugs are not metabolized and they will cause a buildup. In traditional treatment decisions in pharmaco studies or pharmaco research is pretty much a guessing game. You can start with a particular drug dose or particular drug type, and you give it to somebody who has a particular syndrome. That drug may or may not work. It may produce an adverse phenotype. It may be the wrong dose, and finding the right balance between the drug, the dose level, and the individual is kind of an ad hoc or a guessing game, if you will. If we were able to find out how people can metabolize drugs, then it would probably be more efficient for the clinicians to more accurately prescribe drugs that are appropriate for the individual's drug metabolisms. And that's what pharmacogenomics is trying to do. It's trying to link the genotype of particular drug metabolizing enzymes and drug kinetics enzymes and other proteins, and link that to their reaction or their response to a particular drug. And knowing what the background of an individual is, a background of their drug metabolizing enzyme activity is, they can then adjust the drug type or the drug dose to more accurately fit the effectiveness of that drug, and more importantly, prevent adverse drug reactions that might be resulting from an overdose or the wrong drug being administered. So pharmacogenomics is another aspect of predictive genomics, which can have a direct impact on health costs. This has been looked at pretty extensively, and the data that I have here is several years old by now, but it's showing two different graphs. On the top graph is the effect of a pharmacogenomic program in a treatment model. And what it shows is that in the green areas are that the pharmacogenomics testing and adjusting the drugs based on pharmacogenomic information can either be cost effective or cost saving if you include the cost of the genetic test in the overall costs. That's the top figure. In the bottom figure, the pie chart shows that if you ignore the cost of a genetic test, then you get an even more drastic cost savings and cost effectiveness. At least 75% of the number of cases that can be affected by a pharmacogenomics decision wind up being cost effective or cost saved by making use of a genetic test. Now, as I said, this data is kind of old. It's about 5 or 6 years old by now, and the costs of genetic tests have decreased, and therefore, the economic benefits of pharmacogenomic tests are even more extreme now, or more beneficial now. So another way that this was illustrated was with the Kentucky Teachers Retirement System. This is a retirement program for teachers in the state of Kentucky that was set up. And one of the things they wanted to be able to do is, in this retirement system, they wanted to be able to control their healthcare costs. And so they looked into a pharmacogenomics program to see whether or not the pharmacogenomics testing of the Kentucky teachers would help them control the health costs of these individuals. So they had 36,000 individuals enroll voluntarily in this program. They did pharmacogenomic tests on them, and they analyzed the outcomes of these pharmacogenomic tests with the individuals and their doctors and prescribing agencies. And what they found was that the knowledge of the pharmacogenomic information had approximately 30% reduction in risk, adverse drug effects, and reduction in hospitalizations, and even a 25% reduction in slips and falls if people found that they were, maybe more affected by a drug in say a debilitating way. Overall, they found there was a 15% reduction in the healthcare costs of this organization and a 14% reduction in pharmacy savings. So as a result of this program, even though it was not a universal enrollment, they were able to have a significant reduction in the costs for their members in this program. And in fact, at Thermo Fisher, we don't just talk the talk, we walk the walk. We implemented a similar system for Thermo Fisher employees a couple of years ago. And I don't have numbers for this, but I will say that it has been very interesting to see how our costs and the relationship between what we have in our genome and what we have for our medications get affected. So I've been focusing this talk mostly on talking about the link between genotype and risks and pharmacogenomics. One thing I haven't mentioned is that a large portion of the data obtained by the UK Biobank, the FinGen project, the Million Veterans Program, the Taiwan Medicine Initiative, these were all done on modern microarrays made by Thermo Fisher Scientific. This is because Thermo Fisher's had a long history of making these chips. And I say Thermo Fisher, this was actually Affymetrix had the long history, but Thermo Fisher acquired Affymetrix in 2016. So Affymetrix had a long history of producing these chips. The first arrays were begun in the early 1990s. They continued to use the silicon lithographic manufacturing process over the course of years to increase the number of loci and the number of probes that are available on a chip. In 2000, they started introducing software that helped analyze this data. They continued to increase the output of these chips and customizing the chips into SNP chips, gene expression chips, and other types of genotyping microarrays. In 2009, they introduced the Axiom system for analyzing microarrays in bulk. These were 96 sample analyzing plates where you could look at 96 samples with over 500,000 features on a chip per those 96 samples. So it increased the throughput of analysis greatly. Continued to refine this, and in 2013, the UK Biobank Axiom Array was introduced as part of the UK Biobank program, and they've continued to use the UK Biobank chip to collect the data that I've described for you. The Precision Medicine Research Array and the PharmacoScan Array were introduced in 2016 to help with specific alleles that are important in medical syndromes and in pharmacogenetics. We've continued to introduce arrays for the Million Veterans Program, for the Taiwan Precision Medicine Initiative, for the FinGen project, and we continue to refine the protocols, the chips, and the number of samples that are available on a chip to have the utility of these high throughput and many sample projects be useful. The PharmacoFocus Array is available where it analyzes some of the most common drug metabolizing enzyme alleles. And so it facilitates very high throughput pharmacogenomic studies. So today we are really the world leader in some of these predictive genomic studies worldwide, because of the utility of the arrays, the customizability of the arrays, and of the ease of these workflows. So why has the Axiom arrays for predictive genomics been so well utilized? Well, for one thing, the workflow is automated and very simple, and as I've mentioned, amenable to high throughput applications. Per sample, it's very cost effective because it can get a very large amount of data very efficiently. And because the workflow is automated, it can free up staff time for other tasks. And the data output and analysis is very straightforward, using customized and specific software for analyzing the results for this. And finally, for pharmacogenomics, the star alleles that are commonly seen and sometimes a problem for PCR-based assays can be accommodated in the pharmacoscan arrays. One of the important things about the microarray solution, and one of the reasons that it has been used so often for these countrywide or ethnic-specific or microarrays, is that it's customizable. You can tell us what type of alleles you want to have on your chip, and we can make it for you. There's no project that's too big or too small for us. And then we have sophisticated imputation designs that allow the array to be used most efficiently because, by using imputation to increase the number of alleles that can be tested. And as I mentioned, there are multiple testing formats, from individual chips to 96-well to 384-well Axiom microarrays. And then I also want to mention that Thermo Fisher is not just a provider of these tools. We want to be your partner for these types of studies. We have over 30 years of delivering reliable and comprehensive microarray products and end-to-end solutions for laboratories. We have a service lab that can run samples for you if you don't have the capacity to do it in your own labs. And finally, we are here to consult with you to ensure that you are able to succeed with your projects. We've worked closely with the UK Biobank and the FinGen and the Taiwan Precision Medicine Initiative to optimize their arrays, to refine them and make sure they're getting the best results for their needs. And we are willing to do the same thing for anybody else. So please come and talk to us. Let us know what you would like to do, and we can help you do it. When you're thinking about implementing a predictive genomics program, there are a couple of steps that we would recommend for you. First of all, you want to make sure that you have a population-specific strategy and you have access to samples through a clinical care research infrastructure. Again, the Taiwan Precision Medicine Initiative and the UK Biobank have access to state-run healthcare institutes, and this sort of thing is very helpful in being able to uniformly be able to collect and manage information. You want to make sure that you get participant consent for this type of a study. And that's pretty standard, but it's a very important part of ensuring the success of these studies is if the participants know that they are participating in this important research. And then once the data is generated, we have algorithms that help crunch the data and generate results. And then we can go back to the individuals, provide the risks and possible interventions. And we want to continue to make informed recommendations and provide performance metrics for the program so that your predictive genomics program continue to be optimized and made better. So just in conclusion, I will tell you that population-level genetic testing can lead to cost reductions in community healthcare programs. National biobanking collections are becoming a go-to resource for health research, both by the state-run organizations and because they make it freely available for individual and private organizations as well. The pharmacogenomic analysis can reduce the stress on individuals by reducing adverse drug effects and reduce the overall health costs by making the drug dosing and possible side effect causes minimal. Finally, Thermo Fisher Scientific has over 30 years of microarray research and development expertise, and we have the ability to provide efficient and cost-effective solutions for predictive genomics at a scale that is most appropriate for your problems and your needs. Again, please talk to us. We're here to help you do the best science that you possibly can, and we want to be your partner for these types of studies. Finally, I thank you very much for your time, and I'm happy to take questions.
All right, let's get started on our live Q&A. As a reminder to our audience, how to submit questions, just type them into the Q&A box on the bottom left and click submit, and we will answer all of your questions that we have time for. Okay, Steve, we got quite a few questions coming in here. Let's get started. Let's go with this one. Who would be interested in pharmacogenomic information?
So I think basically anybody involved in healthcare should be interested in pharmacogenomics. More specifically, I think that first of all, the patient would probably have the most to benefit from them because they're the ones that are going to have the adverse drug effect, or the lack of effect. If a drug is dosing is not effective. So I think that first of all, the recipient of a drug is probably going to be the most interested. The clinician, of course, is going to be interested in trying to provide the best solution for the patient. So they will want to know what type of drug, what dose of drug is going to be the one that is going to give them the best results, in treating the particular disease or syndrome the person has. Pharmacists would be interested because they would have a very keen interest in being able to help the patient and the clinician, find the right combination of the drug most cost effectively for them to make sure that the patient is helped in the long run. So pharmacists would have a great interest in them. Insurance agents, or it's not agents themselves, but insurance agencies, insurance providers would have an interest because if you can reduce the cost of their health providing, that is going to be, of course, of interest to them. For example, if you can reduce the amount of adverse drug effects, which can lead to external or extraneous hospitalizations or other kinds of problems, the insurance companies would stand to benefit from that as well. And then finally, kind of in a bigger context, the national organizations that provide healthcare for their citizens, like the UK or other countries which have a national healthcare system, they would be interested in this as well because that would lower the costs for the same reason they lower the costs for private insurance companies, lowering the adverse drug effects, or lowering, or more quickly honing in on the drug dose that is going to be most effective for alleviating the problems would be beneficial. So yes, I think all those people would have an interest in the pharmacogenomics.
Very good. Thank you, Steve. And I am noticing that we're having a little flash with your webcam. I'm going to turn your webcam off real quick, and I'm going to turn it back on to see if that helps. Yeah,
I tried the same thing. It didn't. So sorry. Yeah. We'll just have to either go off camera or deal with flashes.
Well, let's deal with flashes. We want to see your face. And if it's, let us know if that's too distracting. But I do want to comment. We have a member in the audience who just said, I'm a pharmacist here. I absolutely love this. So thank you, Tanya, for your feedback on that. Steve, let's jump with this next question. Are other populations targeting predictive genomics?
Yeah, so building off of what was learned from the UK Biobank, Million Veterans, the Taiwan Precision Medicine Initiative, there are a lot of other countries and populations that are interested in this. Thermo Fisher is working, for example, with the Qatari Precision Medicine Initiative or something like that. But the Qatari government is interested in trying to collect information on Arabic populations. And like in Finland, like Estonia, some of these Arabic populations have been isolated, and the allele frequencies in some of the people that are in the Arabic countries are slightly different, and certainly different from what might be in the UK populations. Another one is Korea. They're very interested in identifying the allelic frequencies in some of their populations as well. So yes, there are a lot of other organizations, a lot of other populations that are using the Thermo Fisher model to understand the genetics of their populations.
Very good, Steve. let's go with this question. Could you provide insights on how Thermo Fisher's microarray technology compares to other available options in terms of performance, specifically focusing on its advantages such as improved accuracy and reduced error rates?
Yeah, so the Thermo Fisher, the Axiom platform has been designed specifically for these types of studies. So that we have an extensive array of bioinformatics members, which are very carefully designing the probes to fit the questions that are being asked. This helps reduce some of the false positives and false negatives that might come up. We have redundancy built into the chips that help improve the accuracy of the calls, so that the number of false negatives or false positives is reduced because of the redundancy of the probes that are on the chip. The software has been refined over the course of years to really kind of be optimized to make use of any kind of anomalies that are seen on a chip, helping further to reduce the signal to noise, increase the signal to noise ratio, and reduce some of the visual artifacts that come up. And I think that's just some of the ways in which we are increasing the accuracy and providing a platform that is very accurate for making these calls. One other thing I want to mention is that the probes are designed to be imputation optimal, so that you can make use of imputation to increase the density at which the alleles are being analyzed on the chip as well. So we make our chips, we call them imputation aware, so that the ability to make use of information that might not be exactly on the chip, but that information is important and is easily accessible using our chips. So yeah, we have a lot of things that are built into the way that we design our chips to increase accuracy and usability of them.
Thanks, Steve. Let's go with this next question. Hello, I'm a medical student and I want to specialize in genetic diseases. Is it interesting for my patients to know about pharmacogenomics and worldwide studies?
Absolutely. I think that the more that we can educate patients, pharmacists, and clinicians about the benefits of pharmacogenomics, we all will benefit, because it will reduce the costs and alleviate some of these, like I said, adverse medical effects here. So absolutely, I think that spreading the gospel, if you will, of pharmacogenomics is going to be beneficial to anybody that has an interest in healthcare.
Thank you, Steve. Next question. What are the ethical considerations and challenges associated with the use of microarrays, such as privacy concerns and data sharing?
That's a really good question, which I am not the best person to answer for that. I know that we have our system, our data, our software is designed to be HIPAA compatible, so that we don't we are compatible with all the different kind of laws that are available for keeping healthcare private. We don't make some of the specifics linkage of individuals to the data on our platform. I know that some of the countries that are making use of this information keep it anonymized and don't link individual genotypes to either the results of their studies, unless they ask for them, or for the general population. So you can't link a particular genetic outcome to an individual. I think that's about all I can say about that, because unfortunately I'm not an expert in the privatization of these studies. So I can't address that very deeply. I'm sorry.
Right. Well, I know that your system runs through their organization's HIPAA compliance structure. And so if their institution is safe, then Thermo Fisher's plugs right in.
Okay. Thank you.
Excellent. Steve, this question has come in a couple times, different versions. So I'll add the little here. So can Thermo Fisher really customize any size panel, even a smaller panel?
Yeah, so as I mentioned in the talk, we're here to help you do your science. And if that science is a small scale, we'll help you with that. If it's a large scale, we'll help you that too. We don't have any minimal sample size numbers or minimal number of probes that are on a chip. If you would like to build something, we will help you build it, and we will help you design the best way to build it. So we have a number of different customization options or different customization paths that we can follow for this. We also have, I mentioned this as well, our microarray service facility can also make use of your samples and run them on our provider site, provide you with the data if you don't have access to instrumentation at the scale that you need. So again, the answer to the question is yes. That's the short answer. We want to be your partner. We encourage you to talk to us, talk to your sales rep, talk to your FAS, talk to your young scientists that can help you out with this, and let us help you design the solution that's best for your problem.
Great. This question coming in from Sandra. For predictive genomics, is it better to use microarrays or NGS?
I don't think better is quite the right words to use. Certainly, next generation sequencing is an outstanding platform which provides a tremendous amount of data, and it certainly is possible to use this for predictive genomics paradigm. The issue with next-gen sequencing, though, is that you get tremendous amounts of data, and oftentimes, especially dealing with population-based studies, that tremendous amount of data is difficult to manage, it's difficult to store, it's difficult to extract and analyze the data from it. So I think that next-generation sequencing is outstanding for some things, for discovering new alleles, for example, in small numbers of samples. But for looking at the overall population, I do think that microarrays might be a more practical solution to next-generation sequencing because the workflows are a little bit easier, the data is a little bit easier to manage, and it's just easier for people to actually run the experiments and look at the answers. So next-gen is ideal for some things, and the microarrays are ideal for things as well.
Thank you, Steve. This next question is two parts. So I'm still not clear about why microarrays are being used instead of NGS. Can you reiterate why arrays were chosen for population studies?
Yeah, I think that links to the answer that I just gave. It's because the microarrays can handle a large number of samples very efficiently and very practically. The amount of data that comes off of a microarray is not as great as next-gen sequencing. So handling this data is easier on a very large scale, on a 100,000 to million sample scale. Storage of the information, extracting information, analyzing the information is easier with a microarray than next-generation sequencing. We can run a large number of samples at one time. Like for example, for the pharmacofocus, I mentioned that is on a 384 well plate, so you can analyze 384 samples at one time in a matter of a couple of days using that platform. So you can generate a lot of data very quickly that is specific for the question that you're answering. Next generation sequencing is fantastic for generating a vast amount of data. But the drawback to that is you have a vast amount of data. And so you have to store that, you have to analyze it, you have to extract it, and it's just not as practical in some cases.
Steve, how can I get in touch with someone to create a predictive genomics program?
You could email us at Thermo Fisher Scientific. You can contact myself or some of the other people here on our website. And we'd be happy to help you with that as much as possible. There's also a survey that I think is associated with this talk that you can fill out that with your contact information, we'll get in touch with you and assist you any way we possibly can.
Yes, and to piggyback on that, your survey is located on the far left of your screen. There's a couple questions, and below that are some resources that can help you with your predictive genomics journey. Steve, it looks like we do have some more questions, but it looks like they'll require a little bit more follow-up. So let's wrap with that one. And do you have any final comments for our audience?
I think the last thing, we presented a lot of information here to you, and there's a lot of publications coming out. You can look at the specifics that way by looking at the literature. I think the most important thing is that Thermo Fisher, again, has an interest in you getting the best science for yourself and for your customers, if you have them, for your patients, and that sort of thing. So we want to emphasize that we're your partner and not just a provider of tools. And we want to help you do the best you can for whatever question, whatever people you're trying to help out. So please talk to us and let us help you.
Thank you again, Steve. It's always a pleasure having you present for everybody with your excellent information and your insight. We do want to thank Labroots and our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Now, before we go, I do want to thank the audience for joining us and for their interesting questions. Again, there were quite a few questions that we're going to follow up on, and any of those questions that come in during the on-demand period, they will be addressed via the email address you provided at the time of registration. Today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you everyone for joining us. Take care and have a great day.
Thank you.